Chapter 11, which is about platelet production, the structure and function of platelets. We sort of covered that a little bit in Heme 1, but we're going to go in a lot more depth this time. So we're going over all about the platelets, how they're made, and the structure, and all the functionality of the platelets. So it starts off as a megakaryocyte, or megakaryoblast. So it's a colony-forming unit, and it eventually will grow. And the cytoplasm is a key for the platelets. So a normal platelet count is roughly 150 to 400,000, give or take. And remember, this is very important in primary hemostasis, because once the subendothelial is exposed, now collagen's there, and that's when the platelets will initiate adhesion and aggregation. So normally a megakaryocyte can shed, you can see 2,000 to 4,000 platelets a day, and the lifespan for a platelet is of 8 to 9 days. So the megakaryoblast, let's look at the next picture here. The megakaryoblast, which you can see a picture of it there, that's usually just in the bone marrow. Almost all of these are in the bone marrow except for the platelets, of course. But the megakaryoblast, you can see they've got what they call cytoplasmic blubbing. It's like these little blippy things all around the nucleus of it. And as the cell starts to mature, it goes through what's called endomitosis. In other words, it doesn't split into daughter cells, but the megakaryoblast actually will duplicate itself inside of itself, because it's the cytoplasm is what we're after. So there's different types of megakaryocyte stuff that we're looking at here. The pro-megakaryocyte and regular megakaryocyte, and you can see the CD markers that go with it also. So to identify any problems with megakaryocyte, like stem cell, in other words bone marrow kind of stuff, we can do flow cytometry, FISH, or special staining. It's called immunostaining, and actually is bone marrow biopsies that we take from the tissue and figure out from that. So you can see the markers from the megakaryocyte. Stem cell one for myeloid is like CD34. Immunologically, we find it CD36, 42, and 62. So what controls megakaryopoiesis? Usually it's hormones of like thrombopoietin, which is made by multiple different organs. Liver is the primary one that's there. Erythropoietin has a tiny bit of effect onto it, but interleukins, and you can see this interleukin 3, 6, and 11. There's also other ligands that are there. There are some synthetic types of thrombopoietins that we have out there, and they're used to treat ITP, which is idiopathic thrombocytopenia, to help elevate the platelet count. You can see those listed there also. So we have to control sometimes that we don't want too much platelet production. So we can inhibit the megakaryocyte growth in vitro, and will also tell us in vivo, by platelet factor 4, the beta thromboglobulin, the neutrophil activating peptide 2, and interleukin 8. That sort of controls so we don't go chaotic with too many platelets. So this is a typical platelet. These are a little bit not typical. These are bigger. But a normal platelet is roughly 2 microns, 2 to 3 microns in size. The MPV for it is usually 8 to 10. They're usually small, circular, might be a little irregular. They do have little bumps, granules, we call them alpha, and dense granules that we'll see in it. Again, the normal platelet count is 150 to 400,000. And the spleen holds about a third of the platelet count that's within the body. So the ones on the right are actually what's called stress platelets. They're actually called reticulated platelets because they are bigger and fluffier and have more granules inside of them. So they are actually carrying chunks of cytoplasm, because remember, they're not a true cell. So they actually have ribosomes. They do have some endoplasmic reticulum inside of them. We do have instruments, not around here, but reference labs, huge labs, they can actually measure the reticulum, the stuff inside of the platelets, which gives us a better idea. I mean, it's real important when you're doing stem cell stuff and when we got tremendous platelet abnormalities. So this is the structure of a platelet. So it has its own platelet membrane, sort of. So it has phospholipids, which, support how the platelet becomes activated, makes its shape change, and that's usually supplied by arachidonic acid, which is located inside of it, which we'll see in the next couple slides. So it is a biphospholipid sort of kind of membrane to it, so it's similar to some other stuff. It does have glycoproteins, which are very important because there are all kinds of receptors that we're going to be talking about here, which are very important on how we go through adhesion and aggregation. So it does have a glycocalyx, so it will absorb stuff. That's how the platelets, when they go through the shape change, so they can have better adhesion and better aggregation. So usually a platelet has a negative surface, so it doesn't stick unless it's activated, because we don't want them activated unless we absolutely have to have them. So here is a surface connected canicular system, and this is sort of resembles somewhat like an endoplasmic reticulum, sort of. The dense tubular system is better because the surface, in other words, it's like a sponge, and it allows stuff, like a bone marrow, allows stuff to get out from inside of the cell, because this is where the alpha granules will secrete out to allow us to have more adhesion and more aggregation. So the dense tubular system, and you can see it from the pictures there, it's like a rough ER. It does have stuff that we need for adhesion and aggregation. It has calcium, it's got cyclooxygenase, it's got enzyme like thromboxane synthetase in it. So it has all the stuff we need. So in the platelets, to maintain its normal disc shape, there are microtubules and microfilaments. It's got the normal kind of stuff like you would think in the cells, but remember, this isn't a true cell here that we're talking about. So you can see the picture on the left is a resting one. On the one on the right is starting to make its shape change, where you get what they call pseudopodia, sticking off. So it's like a paint drop kind of effect is what we're looking at here. So inside of the platelet, there are platelet granules. There are dense granules called delta granules or alpha granules. The alpha granules contain some factors, factor V, von Willebrand's, fibrinogen, fibronectin. They're very important, again, in adhesion and aggregation. Of course, the dense granules are very important in activation too and help with adhesion. The dense granules have ADP, ATP, and you can see them listed there, serotonin. Very important for when we go through adhesion and aggregation of the platelet. So here's looking at the plasma membrane receptors. So we have cell adhesion molecules, and you can see them all listed there. There's a whole bunch of them listed there. The integrin, the leucine-rich, the immunoglobulin, the selectin, the transmembrane. So there's a whole bunch there. But what's important for us is these glycoproteins. So glycoprotein 1A, 2A allows us to attach to the exposed collagen, which is part of the subendothelial lining. The glycoprotein 1B95 is important for collagen and or von Willebrand's factor. So that's dealing with more of an arterial kind of thing there. Glycoprotein 2B3A is very big for fibrin and fibrinogen. It is also with von Willebrand's, but is really big for fibrin. Glycoprotein 6 is used for platelet activation and has a collagen receptor. And then we have platelet activation factor, PAF receptor, which helps with the shape change and secretion and the whole thing. And you can see the pictures on there on the platelet of all the different ones there and all the receptors that we have. Other platelet receptors we have is factor V, which allows factor V, of course, to attach, an ADP receptor, which helps with calcium, epinephrine receptor for activation of the platelets, thrombospondin receptor for another aggregation, thromboxane A2 and prostacyclin 1, which are big part of inflammatory kind of things, which help with the calcium release for the dense tubular system, which help with all the different types of granules. Now there's a video here I'm not going to play, but it's very important for you to play it, and we will actually play it in class, but it's an excellent video, explains this in great detail, so you get to hear it from a different way, but a visual thing. Highly recommend you look at it multiple times. So a platelet adhesion, remember this is where we get that initial shape change. So the subendothelial is exposed, allows collagen, which then that collagen will allow us to attach with its glycoprotein receptors. Von Willebrand's is very important. That would be the glyco 2B95. The collagen one is a 2A, 3B, if I remember correctly. That will attach. And that's really important where we have high shear rates, which means that's in an artery because we're pressurized. So we adhere. That's the first thing it does. Again, watch that video. The platelet aggregation allows for more stuff to trigger thrombin to come out. And when that happens, when the thrombin this is now bringing in the coagulation cascade scheme. So we have all these stuff coming out, ADP, calcium, thromboxane A2, everything's coming out to allow for more platelet aggregation to occur. Because when the adhesion occurs and aggregation, secretion happens. And when the secretion comes, that's what allows the aggregation. So in the secretion is alpha in the dense granules. That's where that thromboxane A2 and ADP are being secreted, which allows for aggregation. And then it comes to a stopping point, which is pretty amazing. So you can see some of the receptors there. So remember, in primary hemostasis, we have normal blood flow, right? It's negative, nothing sticks, there's no subendothelial system, everything's moving pretty fast, no activation of any kind. Normal, that's normal, okay? It's nice and smooth. But when we have trauma, where that subendothelial is exposed, and you can see the picture there, that allows the platelets to adhere to the collagen with its glycoprotein receptors, which allows, depending off its intrinsic now involved or extrinsic involved, depends on if tissue factor gets involved or not. And if it's an artery, then von Willebrand's might be activated. So it depends on, where the actual trauma is occurring. So von Willebrand's, as you can see here, actually gives us like another latching point. And you can see the receptor for that, the 1B95, which is the von Willebrand's, to allow it to attach to the collagen. So we got two things to anchor it. And then you can see the blue circular thing in the middle. That's where the platelet is now attached, and it's going to be releasing thromboxane A2 and ADP to allow more aggregation of platelets to occur. So once the platelet is activated and all the aggregation comes in, the fibrin will go through, because now secondary hemostasis is coming in. So the fibrin will go through all the platelets that have been activated and done their little shape change. So we get a nice, stable, strong platelet plug. So the platelet activation pathways, they call them G proteins that actually control how the cell releases stuff from the dense granules. Echinozoid synthesis, which is sort of like an inflammatory process with the platelets. So when we have the platelet adhesion and then we do the secretion, allowing for more aggregation, sorry about that, we have thromboxane A2 and prostacycline, because inside of the platelets is arachidonic acid, and you see that bottom picture. Arachidonic acid is in the cytoplasm, and remember cyclooxygenase is in there. It will convert arachidonic acid to thromboxane A2. That's the TXA2. That will come out and allow for platelet aggregation to occur. Okay, so this is just basic platelets. Again, it is essential, very important, you look at these videos multiple times to understand what's going on. Make sure you read the chapter, look at any questions, and we'll explain this all in lab or in lecture. Thanks.